And then at the very end, I'll remind you about things this week that are coming up. You have 2 things due on Wednesday, and then we're also going to take a look at what's going on next week. So last week, in terms of viral structure and reproduction, we got through talking about viral structure. So we talked about the fact that all viruses have a protein coat that's called a capsid that surrounds their genome. And then we said that, one, the capsid can differ between viruses in terms of their shape. And then we also said that the genome of a virus varies depending on the virus. And then we said that some viruses, and this includes many viruses that infect animal cells, also have what's called a viral envelope that surrounds the capsid. And the viral envelope is actually a membrane that is derived from the cell that the virus infects. So the last thing that we talked about last week was just what happens in each of these steps of a basic viral reproductive cycle. And then I told you that this week we were going to start by talking about the viral reproductive cycle for 2 viruses. One of them is going to be phage lambda. And I didn't write this stuff on the board last week, so I'll write it on the board this week. Phage lambda is a bacteriophage, which means that it's a virus that infects a bacterial cell. And its genome is a double-stranded, this I'm just using that's my own abbreviation, double-stranded, so DS just as double-stranded. Its genome is a double-stranded DNA molecule. Okay, so that's one virus that we're going to talk about. The other virus that we're going to talk about is HIV. And HIV has an envelope. Its genome, has an envelope, infects animal cells. And what we'll see is that actually HIV, it doesn't infect all cells of your body. It actually infects a very specific cell type called helper T cells. And I'll write that on the board later on. And then third thing you should know about HIV is that its genome is a single-stranded, so SS is just single-stranded, is a single-stranded RNA molecule. Okay, so those are the 2 viruses. As I'm describing each stage, the way that I'm going to do it is I'm actually, this slide, which is the next slide, and the slide after it is basically taking you through the steps of phage lambda infection of a bacterial cell. So the first 2 slides after this slide are showing you the reproductive cycle of phage lambda. I'm going to write out all the phases on the board. And then the next 2 slides after that is the same thing, but it's for HIV. So it's the steps of HIV infecting a helper T cell, and then the different steps involved in replicating and then releasing new virus from that cell. Okay, so you can go through a lot of what I'm going to say, or some of what I'm going to say is written down below for each of the steps, but you should probably write down what I put on the board you should write down what I put on the board for this. But anyways, the way that I'm going to present it is that I'll just basically take each stage. So we'll start with attachment, and I'll describe how phage lambda attaches to a bacterial cell, and then I'll talk about how HIV attaches to a helper T cell. So the first stage of the viral reproductive cycle is attachment. And so this attachment is the virus binding to the surface of a cell. So this is in general what happens. The virus binds to the surface of the cell. And for phage lambda, what happens is that the phage has these things called tail fibers. Oops. And the fibers of the tail of a phage bind to proteins that are in the outer bacterial cell membrane. So these would be cellular proteins of the bacterial cell that are embedded in the outer membrane of a bacteria, and the phage lambda tail fibers are going to bind to those proteins that are embedded in the outer cell membrane. Okay, and then HIV is going to do something very similar. What HIV has are glycoproteins in the viral envelope. So let me explain each of those things. So we said that HIV has an envelope that surrounds it, and these glycoproteins, and a glycoprotein is simply a protein that has a carbohydrate group attached to it. So a glycoprotein is a protein that has a carbohydrate group attached to it, and these glycoproteins belong to the virus. So these glycoproteins are embedded in the viral envelope, and they bind to receptors. And in this case, when we're using the word receptor, we're referring to a protein that's embedded in the plasma membrane of the helper T cell. So the helper T cell is the human cell, well, we'll just say it's human for right now, that the HIV is infecting. So glycoproteins in the viral envelope bind to receptors in the plasma membrane. Of human cells called helper T cells. Not too important. You don't need to know now what helper T cells are. We will talk about them at the very end of the course. The last, second to last topic that we talk about is immunology. And helper T cells is a cell type that's part of your immune system. And one of the things that they do in your immune system is they help to activate two other types of immune cells. They help to activate B cells so that they perform their function. And they also help another T cell type by activating them, and then they go on to perform their function in the body. Okay, so that's the attachment phase. The next phase is entry. And the important part of the entry phase is that the viral genome enters the host cell. And so this is going to happen slightly differently. Well, very differently in phage lambda versus HIV. So for phage lambda, it's going to inject its DNA into the cytoplasm of the cell. So what happens in phage lambda is that the capsid is not actually going to enter the cell. The virus just injects the genome into the cell. And then in contrast to that, HIV, what's going to happen with HIV is that its envelope fuses with the plasma membrane of the cell. So remember, the envelope is a membrane, and the membrane of the virus is going to fuse with the plasma membrane. And fuses with the plasma membrane, which releases the capsid and its contents, which is the genome, and also two other things we'll talk about, and its contents into the cytosol, or cytoplasm, whatever you want to call it. So with HIV, the envelope's going to fuse with the plasma membrane. That releases both the capsid into the cytosol, and the capsid at that point is still surrounding the RNA genome of the virus. So one thing I'm not going to write on the board is that, so one of the things that has to happen with HIV is after the capsid is released into the cytosol, the capsid proteins are removed, and that releases the RNA genome. And then it also, the HIV virus, in addition to having the genome encased by the capsid, you also have this enzyme called reverse transcriptase. And this is an enzyme that HIV has that phage lambda does not have. And what we're going to see is that reverse transcriptase is going to be involved in taking the RNA genome of HIV and converting it into a double-stranded DNA molecule. So that's what reverse transcriptase is going to do, and I'll describe that again later on. But once the capsid is removed, it releases both the RNA genome and this enzyme that's contained within the virus called reverse transcriptase. Okay, once the viral genome has entered the cell, what you're going to get is several of the viral genes that are contained within the genome are going to be expressed. Okay, so several of the genes within the viral genome are going to be expressed. So that means that you start to make the proteins that those genes code for. And expression of these genes is going to lead either to step 3, or some viruses skip step 3 and go to step 4. So step 3 is integration, which last week I described to you as a stage where, the genome of the virus is going to be incorporated into the host genome, so the genome of the cell that it's infecting. So it may go directly from stage 2 to stage 3, and that's the integration stage. Other viruses are going to skip the integration stage and go directly to step 4, which is production of viral components. Okay, so you don't need, I'm not going to ask you for that detail that some viruses do stage 3 and skip stage 4. Both the viruses that we're going to talk about do both, so they have stage 3 and also stage 4. Okay, In terms of the integration, the viral genome is incorporated into the host cell genome. And for both viruses, this is going to involve an enzyme called integrase. So the enzyme that's going to perform the insertion of the viral genome in the host genome is called integrase. And that's going to be coded for by one of the viral genes. That part you don't need to remember, but that is coded for by one of the viral genes. And what integrase does, what you need to know for this class is that it incorporates the host, the viral genome into the host genome, but it does that by cutting. So it cuts the host cell DNA, and then it's going to insert the viral DNA into the chromosome of the cell. Okay, so it's going to cut the host DNA and then insert the viral genome into the chromosome. For phage lambda, this step is rather simple. So it's going to involve integrase, and the double-stranded DNA molecule can be directly integrated into the DNA of the chromosome. So remember that phage lambda has a double-stranded DNA genome, and that double-stranded DNA molecule is directly inserted or integrated into the host cell DNA. So remember that the DNA inside of a cell is going to be a double-stranded DNA molecule. So for phage lambda, its genome is also double-stranded DNA, so you can directly incorporate that into the host cell genome. And it's going to be different for for HIV because HIV has an RNA genome, which is single-stranded, and you can't just simply incorporate that single-stranded RNA molecule into the DNA directly. So for HIV, the viral genome, the RNA viral genome, is copied into a double-stranded DNA molecule. And this copying is performed by reverse transcriptase. Oops. Okay, and so I'm not going to ask you about the steps in this, but what happens is so let's say that this is the single-stranded RNA molecule, and what reverse transcriptase does first is that it makes a strand of DNA that's complementary to that RNA strand. So by complementary, I just mean if the RNA molecule has a G here, then this strand's going to have a C there. You're just using your base pairing rules. So reverse transcriptase is first going to make a strand of DNA that's complementary to the RNA. And then what's going to do is it's going to take this DNA strand that it just made using the RNA genome, and it's going to make a second strand of DNA that's complementary to that strand, and then that's your double-stranded DNA molecule. Okay, so those are kind of the brief steps in terms of what's going to happen of taking a single-stranded RNA molecule and making a double-stranded DNA molecule out of it. So in terms of HIV, the first thing that you have to do is copy the genome, the RNA genome into DNA. And then once it's copied into DNA, then the double-stranded DNA is inserted into the host chromosome. Okay. You guys have any questions so far?
So the reverse transcriptase, like, how does it have access to timing? Or is the timing like all around it? Like,
It's in the cell. So basically everything that's needed. So one of the things that you probably don't remember from last week is that viruses don't have ribosomes, and they also don't have most of the machinery that's involved in replicating the genome. And then I don't believe there's any cell that has reverse transcriptase. I think that's specific for viruses. But anyways, they don't contain most of the proteins that are involved in actually duplicating the genome. And then all of the components, the building blocks that are needed. So for both the proteins, so the viral proteins that are going to be made, and then also any nucleic acid that's made, all the components like the nucleotides and the amino acids come from the cell that it's infecting. So the cell itself would have amino acids in it because it's using those to build its own proteins. And then the virus just hijacks that and uses the cellular components to synthesize its molecules. Yeah. Okay. Other questions before I move on to the next step? Okay, so step 4 is synthesis of viral components. So this is going to involve the synthesis of two things. You have to replicate. And when you replicate, you're going to be synthesizing a replicate of the molecule. You have to replicate the viral genome. And then also you have to synthesize viral proteins. And that includes the viral proteins that make up the capsid. So you have to synthesize, that'll include the viral proteins that make up the capsid of the virus that you're going to end up assembling later on. Okay. For phage lambda, so we just went through the integration phase. One thing that's going to happen in this phase is that the viral DNA must be excised. So you're going to cut the DNA back out of the host cell genome. And then after you've excised the DNA, host cell enzymes make many copies of the double-stranded DNA molecule. So this would be replicating the host cell genome. So once you've cut out the viral DNA, so this would include an enzyme that we haven't talked about yet, but that we will, called DNA polymerase. that in your cells is involved in replicating, well, in all cells, is involved in replicating your chromosomes. And so DNA polymerase would basically, the phage lambda would hijack that particular enzyme for this process in replicating that double-stranded DNA molecule. The host cell enzymes are going to make many copies of the phage DNA. It's also going to transcribe genes. So it's going to take genes, transcribe the genes in the viral genome. Remember that transcription is this process where you take DNA, so it's going to take the viral DNA, use it as a template to make the RNA molecule that the gene encodes. And then after you've made the RNA molecule that the gene encodes, host cell ribosomes. Translate the viral RNA, which you made in the second step here, sorry, third step. Okay, there we go. So then the host cell ribosomes are going to translate the viral RNA into viral protein. So you're replicating the DNA again, and then you're going to use the genes that are encoded within the DNA to produce proteins, and that's obviously going to, just like a normal cell, that's going to involve both transcription and translation. Okay, I don't have any more. Okay, so the other virus, again, that we're talking about is HIV. And for this step, you're not actually going to excise the DNA, so there's no excision of the DNA that you just incorporated into the viral genome for HIV. And what's going to happen in the case of HIV is that the viral genome, the integrated viral genome, is transcribed to produce many copies of the viral RNA. Okay, and remember that so the RNA that you're making in this step is going to be the genome of a virus that you're going to build later. Because remember, HIV has a single-stranded RNA molecule as its genome. So this is the way that you produce the genome that you're going to use to make new viruses for HIV. And then in addition to serving as basically producing the genome for the virus, the viral RNA molecules are used, are translated to produce viral proteins. Okay, and then step 5 is viral assembly. So you have to assemble the capsid has to surround a viral genome, and then in the case of HIV, you're going to also enclose in the capsid a couple copies of reverse transcriptase, which is that enzyme. In terms of viral assembly, we're not really going to get into any details in terms of the differences. So for some viruses, so simple viruses, this assembly process happens, so they're going to self-assemble, so it doesn't take any help from anything, from the cell. But that's not true for bacteriophages and HIV. But one difference that you're going to see is that for HIV, the viral assembly actually happens in 2 steps. So the first step, what's going to happen is the capsid is formed around the viral genome and then also the molecules of reverse transcriptase. And then what's going to have to happen in the second step is that capsid acquires an envelope. But that second part of the virus acquiring an envelope which surrounds the capsid, that actually happens in step 6, which is release of the virus. Okay? So no details about viral assembly that you have to remember. But what you're doing is assembling the viral components to make new viruses. So I'll write that on the board. And when I say viral components, again, I'm referring to the proteins that make up the capsid, and then also the genome that is going to be contained within the capsid. Okay, and then the last step is release. And in this case, the virus is released from the cell. And this is going to happen differently when you're talking about phage lambda and HIV. What we're going to see happens for phage lambda is that you actually have to destroy the bacteria cell. So the bacterial cell is going to break open, and that's going to release the virus for phage lambda. But then for HIV, what happens is the capsid containing the viral genome moves to the plasma membrane, and then a piece of the plasma membrane of the cell surrounds the capsid, and that piece of plasma membrane that surrounds the capsid is going to be the viral envelope then. And what happens is that piece of membrane pinches off to the outside, and then you have this viral particle on the outside that has an envelope which surrounds the capsid, which surrounds the viral genome. Okay, so that's going to be the essential the differences between it. For phage lambda, so one difference between the 2 viruses is we said that phage lambda infects bacterial cells, whereas HIV infects animal cells. And one difference that you know between bacterial cells and animal cells is that bacterial cells, and this includes the ones that that phage lambda infects, most bacterial cells are going to have a cell wall. So one thing that's going to have to happen, because that cell wall is a rigid, strong structure, is the virus has to have a way of breaking that cell wall down or open in order for the virus to escape the cell. So in order for phage lambda to escape the cell, the virus is going to lyse. And that just means to break open the host cell. So lyse just means to break open. You can also use this this side note that not that important, but if you're doing research using cells, a lot of times you'll have to isolate proteins or DNA from a cell, and one of the things that you have to do in order to do that is lyse the cells. And that's exactly what you're doing in that case too. You're breaking open the cell so that the contents spill out, and then you can isolate the molecule that you want from the cell. And this is going to involve an enzyme called lysozyme. And lysozyme digests the bacterial cell wall. Okay, so lysozyme, again, is a viral protein, and lysozyme is going to eat or digest the bacterial cell wall, and that's going to cause the cell to burst open. Okay, and then for HIV, what's going to happen is the capsid, which surrounds the viral genome. So you have the capsid already assembled around the viral genome. That happened in the assembly phase, so the capsid which surrounds the viral genome. Associates with a portion of the plasma membrane of the cell. And then the membrane enfolds around the capsid. And I'll show you an actual picture of this drawn out. So it enfolds the capsid and buds off from the surface of the cell. So when HIV is released from a cell, the cell doesn't actually die like it does in terms of, or die like it does when phage lambda has to be. So the cell dies when phage lambda is released from the cell. So you actually have to kill the bacterial cell. But when HIV is released, you're not going to kill the actual cell. Okay, just that last step for, so this is what they're showing you. So these are the capsid proteins that are going to make up the capsid that surrounds the genome of HIV. And then this is showing you the piece of plasma membrane enfolding around the capsid that contains the viral genome. And then eventually this portion of the plasma membrane is going to pinch off or bud off. And then this is the virus that has an envelope around it. And that envelope, again, that surrounds it used to be a part of the plasma membrane of the cell. Okay, do you guys have any questions about the steps in making, in terms of replication of both HIV and phage lambda? Yeah, sorry.
Once it, like, the virus enters inside the cell, I guess HIV, does it continuously make it then?
I don't know the answer to that. I don't know. I don't know if it continuously makes it. I don't know how much of a lag time there is between infection and actual production of new proteins. So yeah, I don't know. Don't know the answer. Other questions? Okay, so the last thing that we have to talk about, oops, and then we'll take a break. I didn't realize we had gone so long. Sorry about that. Is the fact that for phage lambda, and this is also true for other bacteria. Where did I put their rag? Oh, thanks. For phage lambda and other not all, but other bacteriophages, they can go through what's called a lysogenic cycle. Or a lytic cycle. Okay, so on the board here, this is showing you a bacteriophage, and this is showing you the lytic cycle of the bacteriophage, and this is the lysogenic cycle. So one major difference between the two is that the lytic cycle is going to be the cycle where you actually produce and release new viruses. So in the lytic cycle, you're actually producing and releasing new viral particles. And so in the lytic cycle, that's the cycle that the bacteriophage will undergo where it actually lyses the cell. So in the lytic cycle, you're going to lyse the cell and release new viruses. In the lysogenic cycle, there's no production of new viruses, and you're not going to lyse the cell in the lysogenic cycle. What happens in the lysogenic cycle is that the phage DNA becomes incorporated into the host cell genome. And then what happens is that the incorporated viral DNA just gets replicated as the bacterial cell divides. So every time the bacterial cell divides, it's going to replicate that chromosome. So that's what they're showing you here. They're showing you one cell, and then when it divides into two, both the cells are going to contain the DNA of the virus incorporated into the chromosome. And then what you can eventually do is excise that from the host cell chromosome. And I don't know, in case you're wondering, I don't know the answer to what's going to push a virus from the lytic cycle into a lysogenic, or sorry, from a lysogenic cycle into the lytic cycle. Okay, so that's the last thing that we had to cover. Do you guys have any questions about that?
So they don't happen at the same time. It doesn't switch on.
Well, once you enter the lytic cycle, you're going to destroy the cell. So there's really no going back from that. So once you kill the cell, it obviously can't replicate anymore. So yeah, no, there's no, like once you enter the lytic, there's no going back to the lysogenic. Other questions? Yep.
So the lysogenic is essentially just replicating the viral DNA, and lytic is destroying like cell wall.
So yeah, the second part is that you are going to destroy the cell, but you're going to destroy the cell producing more viruses and releasing them. So I would the way I would put it is that in the lysogenic, the key important difference that you guys want to remember is that in the lysogenic cycle, you're not actually producing more viral particles and releasing them. So in lysogenic, all you're doing is replicating the viral genome along with the host cell genome. That's all that's happening in the lysogenic. Well, it's not maybe the only thing that's happening, but that's mainly what's happening in the lysogenic cycle. But then in the lytic cycle, what's going to happen is that you're actually going to make new viral particles, and then the new viruses that you're going to make are going to be released from the cell. And you're right, in that process of releasing it from the cell, you're going to lyse the cell. So lysis of the cell does happen in the lytic cycle and does not happen in the lysogenic cycle. Okay, do you guys have other questions? Okay, so we'll take a 10-minute break there, and then when we come back, what we'll do is we'll answer the lecture questions for cell structure, the rest of cell structure and function and viral replication, structure and replication, and then we'll move on to membrane structure, synthesis, and transport.